Sometimes, ideas found in books and movies can seem unrealistic and impossible. However, in most cases, the fantasy or science fiction is based on reality and facts. A good example of this is Jurassic Park. DNA found in the stomach of a mosquito preserved in amber is used to clone dinosaurs. Though this occurrence in nature is highly unlikely, it is not impossible. The movie helped to raise the question among the public: could dinosaurs and other historical creatures be brought to the present time? This very question is at the head of the research field that studies ancient DNA. The study of ancient DNA is the study of DNA samples taken from ancient specimens. The samples are recorded, and so far a growing library of DNA can be found for dinosaurs, extinct mammals, prehistoric plants, and even ancient humans. These samples can be found within mummified tissues, skeletal material, preserved tissues in ice and permafrost cores, lake or ocean sediments, and preserved plant parts. The study began in 1984 when a team of researchers at the Smithsonian Institution, located in Washington, found DNA in a quagga, an extinct close relative of the zebra. The particular specimen had been preserved for over 150 years. Its DNA not only found to be intact, but it could be extracted and sequenced. Though the study was approached with much hope, it was not long before scientists found many holes and problems in their work. DNA sequencing was first researched in the late 1800s, and it wasn't until the mid-1900s that the first animal was cloned. The first animal cloned from an adult cell was Dolly the sheep, who was cloned in the late 1900s. The big difference between the DNA used in these experiments and the DNA found in ancient samples is the difference in age. The DNA sample taken from Dolly's biological mother was taken and inserted into an N-nucleated egg cell in a very short period of time. As scientists have found studying ancient DNA, samples are more likely to contain more and more post-mortem mutations, most common of which are missense mutations. Not only this, but under the freezing temperatures in which these samples are preserved, the cells of the animals will burst, damaging the DNA and rendering it useless. These mutations in damaged DNA are the primary obstacle that ancient DNA researchers must face. Despite these challenges, some scientists have devoted their lives in an attempt to sequence DNA from prehistoric species. Advancements in technology, including the polymerase chain reaction, which allows scientists to quickly amplify DNA sequences, have proven very useful in the analysis of ancient DNA, as they are very effective even when using very small samples, like those found in prehistoric organisms. Thanks to advancements in technology like these, the science has once again gained some momentum since its initial start. Mammoth remains have been one of the focuses for the study, and scientists are working on cloning one. Because of the cold climate that the mammals lived in, many of the remains, including soft tissues, were preserved in ice, silt, and mud, though most of the DNA is damaged. Many whole carcasses have been found in northern regions like Siberia. Once found, these remains are excavated and moved to research labs and museums where they can be further studied and analyzed. It has been proven by scientists that an African elephant and an Asian elephant can produce a live offspring, even though the baby did not live more than 2 weeks after its time of birth. The fact that it was able to live for 2 weeks, however, led scientists to believe that a sound reproduction cell from a mammoth could be used to produce a hybrid with a modern-day elephant. Unfortunately, so far, scientists have not been able to retrieve enough genetic material to attempt this. Another possibility is to clone the mammoth. Like mentioned earlier, finding undamaged DNA is extremely difficult, but not impossible, and a team of Japanese scientists believe that they have found a carcass from Russia that was able to keep its DNA preserved for thousands of years. The DNA would have been inserted into an unfertilized egg from either an Asian or African elephant, and, if successful, a baby mammoth would be born. Like mammoths, humans have also been naturally mummified, in bogs and ice or other environments that are exposed to little air or humidity. In some ancient civilizations, like ancient Egypt, humans were even preserved in death intentionally, with the use of chemicals that dehydrate the body. Scientists are primarily using bones and teeth for recording DNA samples, as mummified remains are a much more limited resource. In studying ancient human DNA, cross-contamination can be most problematic, as it would in theory be identical to that of a modern-day human. The study of ancient DNA is not inexpensive. Millions of dollars go towards excavating and preserving remains so that they can be further analyzed in labs. Most funding comes from the government and relies on public interest and support. Though the study has met some resistance, particularly from those with religious beliefs against cloning and evolution, ancient civilizations and prehistoric worlds are far from losing interest, so it is likely that the study will continue to grow and become more advanced. But what can we gain from researching ancient DNA? The answer is truth. The more we learn about our origins and lineages, and the origins of the organisms that we live among, the more we know about the world as we know it today. Insight into evolution will also better our understanding on the topic of extinction, and could potentially help to stop extinction from happening to endangered species today.